Okay, so this is the inheritance patterns of Drosophila Melengaster mutations.
Okay, so to start off with the introduction and the background of the flies, we use the Drosophila Melengaster fruit flies to investigate the Mendelian genetics and the predicted inheritance patterns of the genes. The life cycle of a wild-type fly consists of 4 stages: the eggs, the larvae, the pupa, and the adults. The wild types have a body phenotype that are yellow-brownish with black, but the male and female flies have different features. The males have sex combs, and they have a smaller, rounder, darker abdomen, while the females have a pointier abdomen, and theirs are striped. The objective of this lab was to investigate the generational inheritance of genetic mutations and compare the results to those of predicted Mendelian inheritance patterns of genes. We hypothesized that the offspring would follow the phenotypical patterns suggested by the Mendelian genetics, which for wild types would be 9331.
So background of the single mutant gene we had, which was white eyes. The single mutant gene is recessive. It's located on the X chromosome because it's a sex-linked gene. It is located from nucleotide 270599 to 2796466 on the negatively charged strand. The size of the gene is 5868 base pairs in length. The gene codes for proteins that are called ABC type guanine transporters, and these proteins remove the red and brown pigmentation from the eyes, which results in the white eye color. The protons are believed to be membrane associated and ATP binding proteins that transport pigment into cells responsible for the eye color. The size of the protein is 687 base pairs in length.
So in this experiment, we used the double mutant gene, which is a phenotype of yellow body and white eyes. When we looked at the flies under a microscope, we saw that the females were golden yellow, with the lighter faded stripes on their abdomen and cloudy white eyes, which is what we expected to see. Whereas the males were also more of a golden yellow color, but they still had the solid round abdomen with the slightly lighter brown color. The gene that codes for the yellow body Y is located on the X chromosome from nucleotide 356,509 to 361,245 on the positively charged DNA strand. The length of the gene is 4,737 BP. The gene encodes the protein yellow that controls pigmentation patterns of adult cuticle that covers the body of the fly. And the protein length is 541. Flies with the Y mutation have a loss of gray-black pigmentation that results in yellow-brown phenotype.
So methods for our crosses, our initial trial, we set up 4 crosses between the wild-type flies and the genetically mutated flies. One cross consisted of 2 wild-type virgin females and 4 single mutant males. Another consisted of 2 wild-type males and 4 single mutant females. The third consisted of 2 wild-type virgin females and 6 double mutant males. And the final vial had 2 wild-type males and 4 virgin double mutant females. However, the crosses resulted in the flies died, so we had to reset up the crosses, but due to lack of wild-type females, we had only 2 crosses that we could set up between wild-type males and the genetically mutated females. So the first vial consisted of 4 virgin double mutant females and 5 wild-type males. And the second vial consisted of 4 single mutant females with 5 wild-type males. And then after about a week, when the larvae and pupae were present in the vials were cleared early in the morning, and then after about 7 hours, we returned to the lab and inspected the F1 generation before setting up the F2. Then the F1 crosses were set up for the F1 generation of the double mutant cross. A new vial was set up with 5 F1 males and 5 F1 females. And for the F1 generation of the single mutant cross, a new vial was set up with 4 F1 females and 6 F1 males for the F2 generations.
So methods, the molecular strategies, the purpose of using the DNA extraction, PCR, and the gel electrophoresis. was to see the genotype of the F1 flies from single mutant crosses, as well as also understand the differences at the molecular level between the wild type genes studied in the mutant genes. So the DNA extraction consisted of 4 major steps that included lysis, separation, precipitation, and purification. These procedures were used in the lab to genotype 25 wild type flies, 25 single mutant flies, and 25 F1 single white mutant males, and 25 single white mutant females. The PCR was used to isolate and amplify the specific sequence of DNA that codes for the white mutant gene. The primers used for the wild-type flies were white 1 W1 and the white 3 W3. Primers for single white mutant flies were white 2 W2 and white 3 W2. But for the F1 flies, there were 2 PCR reactions that were different sets of primers. The first reaction used the white 1 W1 and the white 3 primers, whereas the second reaction used the white 2 W2 and the white 3 W3 primers. Gel electrophoresis was used to visualize the fragments of DNA in the wild type single mutant white and the both F1 female and male flies. All 8 wells of the gel combs were filled for the first reaction in the F1 samples. The restriction enzyme was used to cut the DNA, and it was EcoRI. For the second reaction in the F1 samples, the restriction enzyme used to cut the DNA was He11. So our hypothesis for this was to see, we thought that the F1 gel electrophoresis fragments would be bigger than the wild type fragments since they have more DNA, because they are single mutant, so they have a mutant gene.
Okay, so these are F1 generation results versus what is expected. On the left, we have the observed results, and on the right are the expected. So we expected for the single mutant cross there to be 50% heterozygous females and 50% hemizygous white recessive males. And we had 10 total, so we would have expected 5 females and 5 males of those genotypes. However, our results demonstrated that there was 1 wild type male, 2 wild type females, 5 males with white eyes, and 2 females with white eyes. And then for the double mutant phenotype expected results, we also expected there to be because we had 15, we would have expected like 7.5, between 7 and 8 doubly mutated males and 7.5 or 7 to 8 heterozygous wild type females. However, our results demonstrated that there were 2 females with the wild type phenotype, 8 females with the double mutant yellow body and white eye phenotype, and 5 males that were double mutants in their phenotype. So our results were clearly not close to what we had expected.
So for our F2 generation results versus expected, what we expected for our F2 generation single mutant crosses were we expected there to be 25% for all, so 25 males that look like wild type, 25 males that look like white eyes, which is a single mutant gene. Females, 10 of them would be wild type looking, and then 10 of them would be white eyes, the single mutant gene. We clearly did not get the results that were expected. We got no males that look like wild types. Instead, we had 23 males that had white eyes, so they looked like the single mutant gene. We had 7 females that looked like wild type and 10 that had white eyes. For F2 double mutant cross, expectancy was as well as 25%. Well, each of them would be equal. But so there'd be male wild type would be 6.85, normal body and white eyes would be 6.85, yellow body and red eye 6.85, yellow body and white eye 6.85, as well as for females. We did not get that as expected. So for our results, we had 0 males that looked like wild type, 0 males that had a normal body but white eyes, 0 males that had a yellow body and red eyes, and the yellow body and white eyes, there were 23 males. For females, there were 19 ones that looked like wild type. There were none that had a normal body and white eyes. Yellow body and red eyes, there were 4, and yellow body and white eyes, there were only 9, which obviously shows that our genes are sex-linked.
Okay, so these are our chi-square. These are our chi-square results. These are our chi-square results. So as you can see, the observed, we had 19 wild type flies, but the expected was 6.875, which resulted in a chi-square value of 21.38. The wild type body, but white eyes, we had 0 observed, but we had expected a value of 6.875, which resulted in a chi-square value of 6.875. For the yellow body and white eyes, we expected 6.85, but we had observed 32 flies. And for the yellow body but red eyes, we observed 4, but we had expected 6.875, which resulted in a chi-square of 1.2. And when we added all of these values together, we got a total chi-square value of 121.255, which is an extremely high value. And by looking at the chart with the degree of freedom of 3, the p-value is less than 0.01. And this was for the double mutant phenotype results. And for the single mutant phenotype results, there were 7 observed wild type, but we had expected 20, which resulted in a chi-square value of 8.45. And for the white eyes, we had expected 20, but we had observed 33, which also resulted in a chi-square value of 8.45. And then when we added those together, we got a total chi-square value of 16.9. And with the degree of freedom of 1, the p-value was also less than 0.1. So our results are clearly significantly different than what we had predicted them to be based off of the Mendelian genetics pattern of inheritance for sex-linked genes, which means that we would reject our hypothesis that the genes are following the pattern of Mendelian genetics.
So for our discussion, as obviously was discussed before, our hypothesis was wrong. Our sex-linked genes did not follow the Mendelian genetic phenotypes. So each cross resulted in a high chi-square value that corresponded with a low p-value. This indicates that the results were significantly different than the results expected from the patterns predicted of the Mendelian genetics, which is what we talked about. Due to the significance difference, we rejected our hypothesis that the offspring would follow the patterns of the Mendelian genetics, as the results were significantly different than predicted. There's a variety of potential areas that could have contributed to these unexpected results, such as if the females were not virgins or if all the adult flies were completely cleared from the vials. We also had lots of trouble trying to get our crosses to work, so probably next time, to avoid so many errors and see better results, we could make more crosses. We could set up more crosses for the future and be able to not run out of wild type flies or wild type females that are virgins. Anything else that you want to add to our?
Yeah, just to go off of the errors. So just to explain them a little further, if the females weren't virgins, they could have already mated with males that we had not controlled in the experiment. Like the female wild type could have mated with another, or the female single mutant or double mutant flies could have mated with males other than the wild type, which we tried to control, which would have resulted in the differences in phenotypes that we noticed. And I think that's pretty much it.
And then our references.
Our references. That's it.